Thank you very much for the introduction, David. And yeah, and I think that actually that's a nice way to introduce me by mentioning that quote, because I think as you'll see in this talk, I'm very interested in kind of all of the novel types of data that we're getting in neuroscience, and I find that very exciting. But I think that theory is really critical because, just getting all these data without developing theoretical frameworks to make sense out of them isn't going to help us very much. And so today I'm going to tell you about a theoretical framework that I've been working on with a senior scientist in my lab named Tirtha or Tirtha Biswas. And Tirtha is in the audience today. And it's called a geometric framework to predict structure from function in neural networks. Now, I'm very motivated to try to understand these large-scale recordings that are becoming increasingly common in neuroscience. So on the right, for example, you're seeing a movie of the larval zebrafish brain, and you're seeing a bunch of neurons flash. In this particular case, it's just spontaneous activity, and you can see there's a lot of spatial structure to this activity, and there's also a lot of temporal activity. And we want to kind of understand, how it is that that emerges mechanistically. And many of us think that the neural network structure is going to be really critical for this. Neurons will be connected in different ways, and then somehow all of this kind of function and activity will emerge from that. But to make sense of these two types of data sets and link them together, we really critically need circuit models that kind of explain how, for example, activity could follow from circuit structure, or for example, how the circuit structure could lead to some particular function. But I want to emphasize that it's really quite challenging to actually make this link rigorously and quantitatively. So if you just start out with the circuit structure, so for example, the connectome of a fruit fly, or on the left, you're seeing a bunch of EM reconstructed neurons in the zebrafish, and you just try to simulate that network, then what people have found is that it often doesn't kind of automatically give rise to function. And I think that this makes sense because individual neurons have a lot of biophysical complexity that might be neglected. Synapses have a lot of complexity. And also all you really get from the connectome is synapse counts, and how that relates to weight isn't 100% obvious. But it's also very hard to go the other way. And so, for example, that's what I'm going to be emphasizing in this talk is, well, what can you say about the circuit structure from the function? And then here, the well-known problem is that you can develop a model that would do a good job at accounting for patterns of neural activity in a data set that have little to nothing to do with the anatomy. So I want to start by just kind of motivating kind of in a cartoon form, well, what it is we're going to do in this talk. And then I'll kind of get into all the details in a few slides. So what we're going to consider is a recurrent network. We're going to have some population of inputs that drive some other populations of neurons that are recurrently connected to each other. And we're going to try to understand how this network could generate different types of input-output pairs. And we're going to have in mind calcium imaging data. And because calcium imaging is much more slow than the kind of changes in neural activity at the level of voltage or firing rates, what we're going to do is we're going to focus on steady states, where we're going to say we have some pattern of inputs that leads to some pattern of outputs, and we want to understand how that pattern of outputs emerges mechanistically from synaptic connections in the network. We're going to do this in a recurrent network of threshold linear neurons. And the reason we're going to do this is that this is going to allow us to have an analytical handle on how the connectivity between neurons from the inputs to the driven neurons or between driven neurons actually gives rise to these functional properties. So for example, each row in this matrix would represent some output neuron. It gets inputs from the different driven neurons as well as from the input neurons. And then there's some color here, which is indicating whether there's an excitatory connection or inhibitory connection. And in this cartoon, then the idea would be that these are a set of weights that would lead you to correctly producing the outputs from the inputs. But the challenge is that this set of weights is not unique. And if you kind of looked for a different set of weights, you could find another one. And if you looked more, you'd find yet another one. And in general, you may actually find an infinite number of possibilities, all of which will kind of give you these kind of steady state outputs from these input patterns. And what we're going to do first is analytically characterize, for the case of threshold linear networks, when the number of response conditions we know about is relatively small, what are all these solutions? But we're going to be very motivated to try to make anatomical statements. And how might we be able to do that despite the variability? So what we're going to say is that, well, we're going to look over the entire ensemble. And if we can find that there are synapses where, for example, the connectivity is always positive and then also always non-zero, then we're going to say that perhaps that would represent a good candidate to make a prediction that there should be some excitatory synapse connecting these neurons. And I think philosophically, part of what I like about this is that, it gives us a way of kind of testing whether this whole modeling architecture of thinking about kind of input-output transformations is giving us a reasonable way of quantitatively linking these structural data and these functional data. Because if this kind of a prediction is violated, it says that there's no choice kind of within this full ensemble where this would be violated. Okay, so I'm going to focus in this talk on this abstract setting, but I think it's just helpful to orient to kind of just talk about one specific example from my past works. So for example, we're interested in understanding how the zebrafish generates optomotor responses, which is a pattern of swimming and turning behaviors in response to moving visual stimuli. And what I'm showing you here would just be, for example, the pattern of visual stimuli shown to the left eye and the right eye, and you might get forward motion, leftward motion, backward motion, rightward motion. And in this past study, we also characterized what were the monocular responses to the different types of motion cues, as well as what were the responses if you put, for example, leftward motion in one eye and rightward motion in the opposite eye. So in addition to measuring the behavior, we found a part of the brain called the pretectum, and that part of the brain gets direct retinal input from direction-selected retinal ganglion cells, and it's interconnected recurrently across the hemispheres as well as within the hemispheres in order to generate binocular responses. So you get direct retinal input of motion from one eye, but then the behavior depends upon both eyes, and the pretectum is the part of the brain that's responsible for doing this. So for example, what we have then would be some pattern of pretectum activity shown either as a shade of color on the left, or for example, with some sort of average fluorescence trace on the right. And what we can see is that across this population of pretectal response types, that you get some amount of responsiveness. So these different rows corresponding to different neurons in this population, some amount of responsiveness to, for example, the binocular motion cue. And for example, if you had some binocular cue, you see that you get more response. If you put in some conflicting cues, you see that some of the responses get lower. You also now start to activate both sides of the pretectum. And for example, if you put just isolated rightward motion, then you can see that you get an opposite, where now suddenly the rightward pretectum is activated. And I also just want to quickly mention that if you look here, you can see that this is an example of a response type that was responding to leftward motion in the right eye, not responding once you added rightward motion in the left eye. So there's some sort of inhibitory effect. But if this signal was isolated, then you can see there's no decrease in the amount of activity. And so this is kind of the origin for why we're going to think about this as some sort of threshold linear response, that we think for explaining these response properties, the threshold really matters. And the steady states you can just kind of manifestly see here that the way that these neurons respond to these conditions is just by ramping up to some level of activity. And we want to try to understand mechanistically how these patterns of activity might emerge from neural network interactions. Okay, so the structure of the talk today is that I'm going to first talk in part one about describing what will be the solution space geometry for this general class of problems. And then I'm going to move on to asking the question from that solution space geometry, are there cases where, despite the fact there'll be variability, some synapse consistently exists across all the solutions? And then finally, in part three, I'm going to generalize both part one and part two to deal with noise, for example, in the neural signals, and show you some numerical tests of the theory that shows that this all works out. Okay, so how do we think about the analytics for the solution space? So we're going to imagine we have I input neurons and we have D driven neurons that are connected to each other in some way. We're going to kind of consider a bunch of different stimulus conditions that are going to be defined by at t equals 0, each of the neurons within the input population transitioning to some sort of activity level, which we'll call x mu1, x mu2, for example, for the different neurons in the input, all the way down to x mu I, that is encoding kind of the pattern of input for the network. In response to some weight matrix we don't know, there's some sort of dynamics, there's going to be some transients, and eventually we're going to kind of approach some steady state activity level. And what we're going to ask is, what can we determine about the weights that are kind of leading you to these steady state levels from these input levels? We're going to do this by assuming a recurrent network of threshold linear neurons. We're going to kind of use the same matrix W to capture recurrent connectivity between Ys and each other, as well as the input neurons from X. And in general, we can have some sort of time constant that would differ between neurons in this network. But since we're going to focus on steady states, that's not going to really matter. And we're also going to specialize to the case where the nonlinear transfer function that determines the firing rate in terms of the input is just threshold linear. And the reason for this is that it's going to allow us analytical tractability, and it's also how we modeled these biological problems in the past. And I think that an emerging body of work is suggesting the threshold linear transfer functions might be kind of a nice sweet spot between biological realism, analytical tractability, and computational power. Okay, so if we now look for the steady states, then because we can set the derivatives equal to 0, all of these differential equations just reduce to a set of algebraic equations. So we have that yi is just now going to be this threshold linear transfer function applied to the inputs of what you're going to get in that particular steady state. And what I want to draw your attention to is that at the steady state level, even though we have a recurrently connected network, for any given neuron in a driven population that I'll specialize to, for example, call the target neuron, the fixed point equation for that neuron only depends upon the ith row of the weight matrix. So what happens is that even though we have a recurrently connected population, at steady state, everything separates into these feed-forward equations, and that's going to simplify the analysis by a lot. Since we're going to be focusing on one target neuron at a time, I'm also often going to drop the index I and just think about a vector of inputs onto a target neuron, and then you would construct the full weight matrix by considering all target neurons sequentially. So then what we can do is that we can say that we've got some number of response patterns. We're going to call that P. And we have for that target neuron, well, what was its magnitude of response into each of these different P patterns? And because we have in mind this threshold linear network, we're going to assume that responses are either positive or 0, and we're not going to have any negative responses of these target neurons. Once we do this, we can kind of combine X and Y together into a matrix, which we're going to call Z. And Z mu M will represent for the muth stimulus pattern what was the mth steady state response of the input neuron, and then WM will now represent the weight onto the target neuron from that mth presynaptic partner. And we're going to introduce N to kind of combine all the synapses, regardless of whether they're from the driven population or the input population. And we're going to assume throughout this talk that we have a limited number of response patterns, in particular, that P is less than or equal to N. Now, the motivation for this assumption is, again, in the larval zebrafish, we generally have a very large number of neurons in these recordings, and we only characterize the network usually under a relatively small number of conditions. But it would be interesting to kind of relax this assumption later and see kind of what would happen as you started to have a larger number of response patterns. And maybe I'll just also quickly remind you, the reason I want to focus on steady states here is because we're going to be thinking about calcium imaging data for which this time course was going to be blurred by the fluorescence responses and the calcium responses. And I'm not going to kind of try to claim that the dynamics we see of the fluorescence signal is governed by this rate equation. Okay. I'm also going to assume that whatever the specified fixed points are, you can make them up yourself, but they're going to be full rank. And because this matrix Z was P by N, and I've assumed that P is less than or equal to N, that's going to mean that the rank of Z is equal to P. I'm then going to extend little z to a capital Z, such that now the rank of capital Z is full rank N. And what that basically means is that for the first M rows of this capital matrix Z, it's just the stimulus response patterns we're trying to explain. And then for the last N minus P rows, they're just going to spend a null space of Z and just complete it so that we have now a complete full rank matrix Z. And the reason that this is going to be useful is that we can now use this capital matrix Z to do a basis transformation from what I'd call physical coordinates, which is the pattern of synaptic connectivity, for example, from presynaptic partners onto the target neuron, into what I'm going to call omega coordinates. And this capital matrix Z can be thought of as a basis transformation. And the reason that this is going to be useful is that once you make this basis transformation, the structure of the fixed point equations become very simple. So for example, this matrix Z for mu equals 1 to P is nothing other than the argument of the threshold linear transfer function in your fixed point equations. And so what you have now is that the equations you're trying to find solutions to all have the form of y mu is equal to a threshold linear transfer function acting on omega mu. And so these omega mu define a convenient mathematical point of view, from a mathematical point of view, linear combinations of weights. Okay, so why are they convenient? Because now we can kind of very transparently see, well, what are the conditions we need on omega in order to get a solution? So anytime that y mu was positive, then the fact that this is positive means that the threshold linear transfer function can't be 0. And so then you just get y mu equals omega mu, and you get exact constraints. So along these so-called constrained dimensions, you can specify exactly what the omega coordinate needs to be. On the other hand, if y mu was equal to 0, then all you get is what I'm going to call a semi-constrained condition. And the reason for this would be that if omega mu is anything negative, then once you pass it through that threshold linear transfer function, it'll become exactly equal to 0, and now you'll satisfy the fixed point equations again. So along these semi-constrained dimensions, what you can see is that the solution space shown here in deep yellow has to have negative values of that particular coordinate. And then finally, because we're considering the case where p is less than n, anytime the mu is greater than p, you get a completely unconstrained condition. And now that can be positive or negative and can adopt anywhere you want within the solution space. And the important thing is, because we now have Z being some full rank matrix, once we specify a solution in terms of these omega coordinates, we can go back to the physical coordinates simply by applying the inverse basis transformation, which in this case would just be this matrix Z inverse multiplied by the representation of the weights in these omega coordinates, which I'm calling omega here. So conceptually speaking, this is a very important slide, so I just will repeat it. So what we do is that we kind of define these convenient linear combinations of the weights called omega coordinates that depend upon the fixed points we want to define. The reason for that is that the fixed point equations become very simple in this basis. Anytime that the output response was positive, we get a constrained linear combination. Anytime it was 0, we get a semi-constrained or sign-constrained combination. And anytime that we don't have a response, because we're in this underspecified regime, we get an unconstrained condition. And we can then enumerate all the solutions in this particular basis, apply the inverse basis transformation, and get back to the original weights. So we now have an infinite number of possibilities. So is it the case that within this infinite possibility, there's any kind of consistent anatomical structure that might be kind of a good target for experimental investigation? So to give you a sense of why this might be the case, I first just want to walk you through a very simple toy example. So let's imagine we have two different stimulus conditions, and we have two different input neurons and one target neuron. So imagine under stimulus condition 1, both of the input neurons are positive 1, whereas in stimulus 2, one is positive 1, the other is minus 1. And then we're looking for weights W1 and W2 such that the target neuron responds with magnitude 1 under condition 1 and 0 under condition 2. Now, if we had a linear network, there'd be a unique solution to this problem, which is shown by this dot here, which would be given by W1 equals W2 equals a half. What that means is that along this constrained dimension, which is fixed by the pattern of the inputs, that you have to go a fixed distance out in order to find a solution. Now, what the threshold nonlinearity now does is that in the other dimension of this weight space, now the semi-constrained dimension, all you know is that the input has to be negative. And so what used to be a unique solution now becomes a ray that extends out. And what I've also done here is I've introduced some concept of the maximum weight bound for the vector. And we're just going to say that biologically speaking, you can't have infinitely large weights. We're going to leave this as an adjustable parameter, but we're saying that all solutions have norm less than some value that we're calling capital W. Okay, so then this is the solution space. So what does that tell us about the anatomy? Well, if we look for W2, even though we have an infinite number of solutions, they're all positive because it was at least equal to a half. And as you kind of explore this dimension, all that happens is the magnitude gets larger. And what that will then mean is that through all the solutions, you're always going to get a positive and non-zero synapse. If we look for W1, in this example, you can see it could be positive, or it could be 0, or it could be negative. But you can imagine that if you decrease the size of this sphere, that eventually all the solutions would lie over here. And then you'd be able to say that W1 had to be positive. So this gives us some notion of W critical, which is the smallest radius of the sphere such that that particular synapse always has a fixed sign for all solutions within that sphere. And then we would say that if it's the case that W is less than this W critical, we would predict an excitatory synapse. An equivalent way to look at this would be to say that, well, rather than kind of changing the radius of the sphere, we can imagine that you just have some sort of magnitude of postsynaptic response, and we can imagine changing that. And what that would do is move this dot out along the constrained dimension. And you can similarly imagine for a fixed radius W that if you move far enough out here, then as you get your semi-constrained dimension, eventually you no longer have an intersection anymore. And once again, you have always have a positive solution. And so then we're also going to define what I'll call Y critical, which if the postsynaptic response magnitude over all patterns is greater than some critical value, then again, you can say that there has to be a weight. Okay, so for illustrative purposes, it's helpful to do one more toy example in 3D. So what now happens if we add an unconstrained dimension? And how do we then think about how to determine whether or not there is or is not a certain synapse? Okay, so what I'm going to do now is I again have one constrained dimension, one semi-constrained dimension, and now one unconstrained dimension. What the solution space will look like is this deep yellow disk, which is all of the solutions within the sphere of radius W. All the solutions are fixed distance along the constrained axis. They all have negative components along the semi-constrained axis, and they can be anything along the unconstrained axis. What I'm also showing on this plot is an orange vector, which points in, it's a unit vector pointing in the direction of the particular synapse that you're trying to ask a question about, of whether or not you do or don't get any variability in synapse sign across the ensemble. And what you can do with thinking about that is it's the vector normal to the separating hyperplane between, for example, W being positive and W being negative solutions. So on this hyperplane, you're then going to get conditions where that weight's exactly equal to 0. So the crux of the problem then becomes, do you get an intersection between this hyperplane, which specifies the weight being equal to 0, and this solution space? Because if you do, then along this intersection, you're going to get exactly 0 weight. And on either side of the hyperplane, you're going to get positive and negative solutions. So in this particular example, you're going to not be able to determine whether the weight is positive, negative, or 0. But if you imagine an alternate orientation of the synaptic direction, then you can have this scenario where actually the intersection to the subspace isn't part of the solution space, because you need to have positive omega coordinates in that semi-constrained dimension. And now here, all of the solutions shown here by that deep yellow wedge are either positive or negative, and none of them are equal to 0. So all solutions have a synapse between this pair of pre and postsynaptic neurons. Now, the reason it was worthwhile to generalize to this kind of three-dimensional case is that remarkably, we have a generalization that kind of now goes into a high-dimensional setting, and we have a geometric reduction that has a very similar form. Now, the assumption we need to make here, and I'll kind of come to this again at the end, is that the stimulus patterns are going to be orthogonal to each other. So we can have as many input synapses as we want. We can have as many patterns less than or equal to that number. But for each two patterns, mu and nu, we're going to assume that the dot product of the neural activity across that is equal to 0. And also we're going to have them being kind of unit norm. And if this condition is satisfied, we can actually exactly analyze whether or not there's an intersection between the solution space in this high dimensional problem and this separating hyperplane in this high dimensional problem. And the way that this decomposition works, oh, and I actually forgot to mention this at the beginning, all of this work is presented on an archive preprint. So if you want more details about what I'm talking about, please refer to this preprint and feel free to shoot me an email and I'd be happy to kind of discuss it more afterwards. But in any case, the way this geometric reduction works is then whatever this kind of high dimensional synapse direction is, we can decompose it into a component in the constrained subspace, in the semi-constrained subspace, and the unconstrained subspace. And then we just need two angles because of this orthogonality condition to specify the orientation of the synapse with respect to these various subspaces. Additionally, because we now have a higher dimensional problem, there's some substructure within these subspaces that matters. And in particular, within the constrained subspace, there's one angle that matters. And the reason for this is because the response direction for the postsynaptic neuron can be thought of as a pattern within the constrained subspace. And then therefore, the orientation of the synaptic direction with response to that pattern is also important. And there's also one additional angle that matters within the semi-constrained subspace. And the reason that this one emerges is because all the solutions have to have negative omega coordinates for throughout the semi-constrained subspace, and then therefore it matters how the synaptic direction is oriented with respect to that all negative orthant. And once you do the analysis, what you find is, again, in the same way I kind of explained to you in these simple toy problems, it turns out that a synapse will exist in all solutions if and only if the magnitude of the postsynaptic responses y over kind of all the different patterns is greater than some critical value y critical. And we also have an exact geometric formula for what y critical is. And I don't want to explain the details of this, but I want to give you a little intuition. So y critical is proportional to w. So what that means is that if you kind of consider larger magnitudes of weights, then you need kind of a larger response magnitude of the activity to kind of exclude all of these kind of the possibility that the synapse is equal to 0. And then there's three constants that depend upon the angles I described to you on the previous slide, one of which relates to the semi-constrained subspace, one to the unconstrained subspace, and one to the constrained subspace. And you got all three contributing to the denominator and just the semi-constrained and the unconstrained to the numerator. So what this means is that if things are kind of good for the experiment, then what can happen is that C can be very large and Y critical can get very small. So this would be a scenario where, for example, the pattern of postsynaptic responses is convenient for the synaptic direction you're trying to determine, and you can kind of suppress Y critical to be low. But on the other hand, if the synaptic direction aligns a lot with the unconstrained or the semi-constrained subspaces, then this thing can approach 1, and then you basically you can't determine anything because you need to be outside of the weight bounds. And what I also want to emphasize is I'm not showing you the form, but there's an interpretable form for C, S, and U, the closed form in terms of this data matrix Z, and all interpretable. And to just give you a little bit of a sense of this, what C turns out to basically be is the correlation in a normalized way between the pre and the postsynaptic neuron activity. So for example, if you're trying to determine connectivity from one presynaptic neuron to that postsynaptic target neuron, and if their response patterns are highly correlated with each other, then you have strong evidence that there may be a connection there, and it makes it easier to determine if there's a synapse connecting between these two neurons. Okay, so this is all well and good, but so far everything I've talked to you about is just exact solutions. So we have a set of fixed point equations, and we ask what are the set of weights that support these fixed points, and is there kind of consistent sign across all of them? But what's missing from this is some accounting for noise. So why is this important? So for example, if we go back to the biological motivation, here I'm showing the average response of one population of pretectum neurons to 3 different stimulus conditions. But you can also see error bars. And this is because there's a lot of trial-to-trial variability. And it's not immediately obvious what is the exact magnitude of the steady state response level. And you can also see for some of these conditions, it kind of is maybe increasing a little bit or decreasing a little bit. So just generally speaking, it's not as though we can look at biological data and say that there is a fixed point at exactly this value. We need to have a little bit of wiggle room and say that, no, we've got some amount of uncertainty in that statement. Once you allow for that wiggle room, the structure of the solution space could in principle change a lot. So for example, here you can imagine a scenario where you look at the error of how well you can predict the output as a function of synaptic connectivity from neuron 1 to neuron 2. And I've drawn an example where the model error is highly correlated. So you can keep the error to be small as long as you change along this direction, where neuron 1 and neuron 2 compensate for each other. And so in general, it could be the case that there could be very different weights within this ensemble that don't fit very well what you'd expect by just looking at the center of this error surface. And then finally, I want to mention that we're also going to be wanting to compare this, of course, to models. And in models as well, we very rarely get exactly the set of fixed points we ask for. So you can imagine that maybe we're going to look for networks that generate this input-output transformation through some sort of numerical optimization technique. And what you might find is that actually the training error doesn't quite go to 0, and it kind of saturates at some level for some period of time. You could definitely imagine if this was you doing this numerical experiment, you'd stop here. So you wouldn't get an exact solution, but you'd have something somewhat nearby. But you can also see that eventually it'll go down, but even then it doesn't go exactly equal to 0. So for all 3 of these reasons, we want to consider a case where we just don't have exact solutions to some set of fixed point equations, but we have approximate solutions. Okay, so then the question I want to ask is, well, what are going to be the neural network weight matrices that lead to fixed points near the specified ones? So to make this precise, I'm going to define what I'll call an epsilon error surface. And what this is going to be is a set of all the weights such that the squared error, meaning the sum over all of the patterns and all of the target neurons, of the prediction of what the steady state level should be for that set of weights, and I'll get to this, what I mean by this a little bit more detail in a moment, minus the target that you're trying to build a model to produce squared is some constant capital epsilon squared. So conceptually, this epsilon error surface is just all the weights with error squared equal to epsilon squared. And because we have a recurrent network that in general could have multi-stability, we're going to define a little bit more precisely what we mean by y tilde mu I of w to be the steady state that you'd approach if the network was initialized at y mu I for all I. So for example, if you have a fixed point there, then you've got an error of 0, and it may be you've got other fixed points elsewhere. Now, this particular notion of the error surface, everything I've said so far has been exact for both feedforward and recurrent, but we're going to need to make an approximation here to kind of treat the recurrent case. And what we're going to say is that let's just imagine that even though we've got a recurrent network, that at the steady state level, all of the presynaptic activity patterns are the fixed points that you specified. Now, the reason that this is an approximation is that once you allow for error, then the presynaptic neurons can move away from this direction z. And if you remember, these z are what define the directions of the omega coordinates in the first place. And so what happens is that accounting for error, you can actually kind of get the geometry itself to wiggle. But we're going to kind of ignore this. And we're just going to say that this is still equal to some sort of threshold linear transfer function acting on the omega coordinates. But nevertheless, when we compare this to simulations for the recurrent network, we'll properly do it in this form that accounts for this source of variability. And we'll just see whether or not this theoretical approximation, we can get away with it. And I also want to mention that this squared error function, when you represent it in omega coordinates, will now have a very simple form where you just need to sum over all the presynaptic neurons the target neurons postsynaptic I, as well as all the patterns mu. You've got your phi of omega I minus yi squared. And notice, for example, that if all the w's are positive, then again, this phi will disappear, and then this is just an equation of a sphere. Okay, so what does this then say about the structure of these epsilon error surfaces? Well, as I just said, if the error is sufficiently small, and if you're in a scenario where all the responses are positive, then for sufficiently small values of epsilon, then the epsilon error surface is just going to be a sphere. Now, what happens as you increase the error? So for illustrative purposes, what I'm going to imagine is that for whatever target neuron we're looking at, we have the p different response patterns, and we're just going to order them in increasing order of magnitude. So for example, y1 would be the smallest response from that neuron, then y2, all the way up to yp. Now, as we increase the error of the epsilon error surface, at exactly epsilon equals y1, we get a topological transition in this error surface. And the reason for that is that all of a sudden, it's possible that at the, all of a sudden it's possible that y1 could actually be exactly equal to 0. And because of that, you get a semi-flat dimension that opens up in the minus omega 1 direction. And then as you increase the error, that infinite line of solutions dilates to become a cylinder. You can now increase the error even further. This cylinder will get more fat. And now at the value epsilon equals to y2, you get another topological transition, where now all of a sudden you can have negative variability along your omega 2 direction. And this will keep going. So for example, at epsilon equals to y3, you'll get a cylinder in the omega 3 direction. But also it gets more interesting now, because it's possible that before you get to this value of the error, at epsilon is the square root of y1 squared plus y2 squared. Now what happens is that you can simultaneously have y1 equals to 0 and y2 equals to 0. And as a consequence, these two cylindrical axes merge into a plane. And now you have some sort of higher dimensional generalization of a cylinder, where rather than having one cylindrical axis, you have two. But what I hope is kind of apparent from this kind of simple example is that if you know what the pattern of the responses are, it's very easy to kind of enumerate what the complete set of topological transitions is, and to then know what the epsilon error surfaces look like for any chosen value of epsilon. And this geometric picture that I'm describing to you here is exact for feed-forward settings where we don't have any recurrence. And we're going to see whether or not the geometry that we have here gives us any insight as well into the recurrent situation. Now, once we now have these solution spaces, so now we won't want to just say, well, what are the exact solutions? We want to say, okay, well, what are all the solutions, for example, with error less than some value? What does the solution space look like there? And this can have pretty dramatic effects on whether or not a synapse has consistent sign throughout the solution space. So let's go now to another simple two-dimensional example. And now let's imagine that, as was the case on the last slide, that for sufficiently small values of error, there's a unique solution. So that's that point there. Now, as you increase the error, the epsilon error surface in general is going to be an ellipse. I've kind of represented it as a circle here just for simple illustrative purposes. And if you kind of ignored the fact you can have these topological transitions, then you would think that all solutions less than value with error less than epsilon would just lie within this circle here. Now, because of the topological transitions, it's possible that a semi-constrained dimension will open up at some given value of the error, and then you get the semi-infinite line that then dilates to become a cylinder. And if you look at this example, what you can see is that because of this cylindrical set of solutions, it suddenly becomes the case that W1 could be positive, could be negative, or could be equal to 0. And if you ignored these topological transitions, or if you ignored this error, you would get that wrong because you wouldn't recognize this kind of large set of solutions here that kind of eliminate your ability to determine that W1 is equal to 0. Now, it turns out that for this case now involving noise, then the generalization of the problem is we don't want to just find a intersection, whether there's an intersection between a zero error solution space and a separating hyperplane. Now we want to look at the separating hyperplane and all models that have error less than some value, capital epsilon. And it turns out that we can also treat that problem exactly in an analytical way, where we can show that there exists a y critical and we can derive an exact upper bound for it, so that if y is greater than this exact upper bound, we also know that it's greater than the true y critical. And in addition to this exact upper bound, we also have an exact lower bound and a leading order approximation to kind of see that these approximations kind of do a good job at accounting for it. But in terms of kind of making quantitatively precise statements, I'm going to be focusing for the rest of this talk exclusively on the upper bound, which then makes theoretically rigorous predictions that if y is greater than this theoretical upper bound for y critical, then we know that it's also bigger than the true y critical. Okay. So with all of this machinery in place, I want to move on to show you that this works and that we can use this formalism now to analytically determine what is the condition for which you need to have a synapse between pairs of neurons, and is this satisfied in, for example, some numerical setting. So I first want to consider a simple feed-forward setting so that we just have a single target neuron, which I'll call Y1. And that neuron Y1 is getting input from I different input neurons. And we're potentially interested in determining information about these different I synaptic weights from the input neurons to the driven neuron. And what we're going to do now is that we're going to analyze the problem theoretically and compute when we think that there should be a synapse between pairs of neurons. And if that synapse exists, what we think the sign should be. And we're going to compare it to numerical simulations that we're going to find using kind of a standard optimization technique. And in particular, we're going to do gradient descent learning. What I'm showing you here is a set of simulations. In each set of simulations, there's both a theory value and a numerical value. The theory value is being tracked by this solid line. And a numerical value are shown in these circles, and you can see it's exactly tracking this line, okay? So what this is saying is that, in this setting where we, again, expect the theory to be exact, as expected, we're able to kind of correctly make the predictions. Okay, so what are these lines? So let's just first focus on, for example, the brown. So what we're going to do is we're going to increase the number of input neurons, or for example, the number of synapses onto Y1. And we're going to increase that along this axis. For each choice of the number of synapses, we're going to generate a random pattern of presynaptic activity and postsynaptic activity that satisfy these certainty conditions, that satisfy these orthogonality conditions, such that n over 4 of the dimensions are going to be constrained in the sense that we should know exactly their magnitude. And then for this one, we're going to have 3n over 4 are going to be semi-constrained, which would mean that n is equal to p, or the number of stimulus conditions is equal to the number of synapses we're trying to constrain. Or for example, in the purple one, we now just again have n over 4 semi-constrained dimensions, so that there's these 0 responses, but now there's also p minus n, or n over 2 in this particular example, null directions where we don't know anything about the response. So we generate a random pattern. Each of these represents one random pattern. We compute from the theory what are the set of synapses that we expect have to be non-zero, and if they are non-zero, what should be the sign. And then we look into the numerical solution that we found with gradient descent learning. And again, because of these semi-constrained dimensions, it can take a really long time numerically to find these solutions for these high values, which is again showing the benefit of the theory. But once we find a solution, we now test the prediction of the theory. We say, okay, well, the theory said that this synapse needed to exist and it should have this sign. We check if that's the case. And if it was the case, we kind of count that as a success. And you can see that again, in this setting, that every single test we performed was accurately predicted for all of these different scenarios. So again, this is in some sense a sanity check because the theory should be exact, and the fact that it accounts for the numerics is comforting. Okay. Additionally, what I want to point out that we find interesting about this, beyond just being able to account for the numerical results with the theory, what we find empirically is that the number of synapses that seem to be certain seems to scale with the size of the system. So it's not as though when you go to high dimensional systems that all of a sudden you can't determine anything about it. And instead, we actually have a theoretical argument, which is in the appendix of the paper, that argues that the number, or what I should say, the probability that a synapse is certain actually only depends upon the ratio of these different types of dimensions. So because we're kind of scaling the number of constrained and the number of semi-constrained with the size of the system, we have a theoretical argument that says that the probability should remain constant, so you should get this linear scaling. And we can't at the moment explain the slope, but nevertheless, we have an argument that accounts for the fact that there is this scaling. Okay, so what about recurrent networks? So here what I'm going to do is a lower dimensional setting because again, it takes a really long time to find these solutions numerically. So I'm gonna have just a handful of driven neurons, I think 4 in this example, a handful of target input neurons, I think 7 in this example. Again, I'm gonna have some sort of random input output configuration I try to produce. And the first thing to just check is that now once we allow for recurrence, do we still have this notion of a constrained and semi-constrained dimension accounting for the structure of the solution space? So now what you're seeing is that each dot in this plot represents one model found with gradient descent learning, and the color of the dot represents the error. So how good that model did in terms of producing the fixed points that we specified. And along the x-axis, we have a dimension that the theory predicts should be constrained. And you can see that along this axis, all of the solutions lie within a very narrow range. And then we have along the y-axis a dimension that the theory predicts should be semi-constrained. And now you see that you get a massive amount of variability along this dimension, just as you would predict from this kind of feed-forward approximation. And at least for this numerical experiment, we don't find kind of any evidence that there's anything really complicated going on in terms of kind of the geometry of this, how the semi-constrained dimension kind of changes for different values of error. We can also then, in a similar way to what we did for the feed-forward network, we can test these predictions. So for each value, each model here, we have some error. We can use our theory to predict what would be the synapses that should, for that value of error, have a fixed sign, and we can test that prediction. And so if you look now first at the yellow versus the red, you can see that as we increase the error, the theory is making predictions of a certain number of synapses that should have a fixed sign and be non-zero. And there's no red X's, which means that the theory never made an incorrect prediction. So that every single time the theory said, okay, for that value of error, I think that these set of synapses should exist and should have this sign. That was always the case within the numerics. And what you can see now in the cyan and the blue would be that if we kind of ignore the nonlinearity and we just kind of treat this as some sort of linear neural network model, then we can see that sometimes this will work, but even for relatively small values of error, we're often making mistakes where it's not doing a good job at predicting which synapses should exist throughout the solution space. Okay, so to summarize what I've shown you, and this is the content of the archive paper. So if you're interested in this, I would really love you to go take a look at it and shoot me or Teotho an email and let us know if you have any questions or comments. But what I've shown you is that we asked when a synapse has to exist to generate some fixed point responses in threshold linear recurrent neural networks. And we developed a theoretical formalism to exactly calculate all of the weight matrices that would work for a small number of fixed points, again, motivated by the scenario we're dealing with in zebrafish. And then we specialized to the case where we had orthogonal patterns of fixed points, and we also derived an exact certainty condition that determined when that synapse was non-zero, and therefore fixed sign across the entire solution space. And to do that, we had to do some geometrical calculations where we checked to see whether the separating hyperplane did or did not intersect with the solution space. We then generalize this whole picture to account for noise, both in terms of finding values of weights to generate fixed points near the specified ones, as well as kind of calculating these intersections. And I showed you that, at least for the examples that we've tried so far, everything works really well. So, I didn't cherry pick anything. So far, the theory just has always worked every situation we've tried it in, and that's good. But I just want to remind you that, ultimately we kind of motivated this with a biological setting, and we remain interested in this. And so I want to just give you a quick taste, which is, this goes beyond the preprint of just where things are going, both from kind of comparing the biological problem as well as kind of generalizing the theory to be general enough to treat the problem we motivated it with. So now I'm going to again go back to this problem of binocular integration in the zebrafish pretectum. I'm going to kind of focus on a subset of conditions that have to do with the binocular integration of leftward and rightward motions between the leftward and right eyes. And I'm going to look at, for example, what are the patterns of synaptic connections that can generate some empirically measured set of steady state response levels in the pretectum. And the first thing we're going to do is just numerically characterize a solution space by just finding a bunch of different solutions using gradient descent learning. And the point that I want to just draw home here is that you can see that there are many points within this matrix. So again, postsynaptic targets in the left pretectum, presynaptic targets could be either on the same side of the pretectum, the opposite side of the pretectum, or the retina itself. We find that there are many synapses that, empirically at least, seem to kind of overwhelmingly tend to be either positive or negative, and then others, of course, that are kind of more split between these two. But the idea of this now being that, I think that this kind of motivates us at least to hope that there could be some example synapses where really there is a consistent connectivity sign throughout the entire ensemble. And also, I didn't mention this at the beginning, but part of what makes this an exciting problem is that there are multiple groups that are combining functional imaging to kind of record these response properties, and then they slice up the brain of that very same zebrafish and look at the EM level anatomy of that system. And so we think that we're kind of ramping up to be able to kind of make some predictions about, well, what might be from this modeling framework, some synapses that we expect to exist that we can kind of compare to that data set once it exists. But this is just numerical. And so what's missing from the theory to allow us to do this kind of fully properly is the assumption that the patterns are orthogonal. And once you kind of allow for non-orthogonal patterns, things get a lot more interesting, but also a lot more complicated. And so this is work in progress that Tirth and I are working on together with an undergraduate student who worked with us last summer and is continuing to work with us now. And actually, Lambus is currently interviewing for graduate school. So if any of you are interviewing him, he's a very talented student, and I would highly encourage you to try to bring him to your institute. But in any case, we're now trying to kind of say, to what degree can we do this geometry? Because again, the solution space analysis didn't depend on this orthogonality condition. All we need to do is figure out whether or not there's an intersection or not. And I don't expect that you're going to be able to kind of figure out exactly what this slide means, but what I hope to impress upon you is that once you start to allow for non-orthogonal stimulus patterns, a lot of complicated things can happen, like the solution space within the semi-constrained subspace, the origin can shift, it no longer has to be circular, it can start to be an ellipse. And because of this, it becomes a lot more technically challenging to determine whether or not there's an intersection, because all the symmetries that we kind of used for the orthogonal case disappear. But nevertheless, we have an approach that I'm confident will exactly work for problems as small dimensional as I motivated here. So we'll be able to kind of do this for the zebrafish pretectum. And we have some ideas, but it's still work in progress about whether we'll be able to push this technique to arbitrarily high dimensions. Because basically the technique we have right now, that again is an exact approach, requires some combinatorial search over some dimensions, which is obviously not going to work for too high dimensionalities. And so anyway, this is just a teaser, but to just say that this is not the end of the story, but we think that kind of starting out with these orthogonal cases and the solution space that we've worked out in general is going to allow us to kind of hopefully make some interesting predictions from the activity levels within the zebrafish brain for anatomy that can subsequently be measured. And with that, I'm just going to stop and acknowledge various people. So first, just the people in the lab. So as I mentioned at the beginning, the vast majority of this work was a preprint that I did in collaboration with Tirtha Biswas, and this has been a true delight. And thank you, Tirtha, for collaborating on this with me. Lambus, I also mentioned, has been helping us once we started to kind of treat the case of non-orthogonal stimulus conditions. And Shelby is my lab coordinator who helps with all aspects of keeping the lab running. And I also want to just make a little advertisement for Genelia Comp Neuro, because I think we have a really nice community. We're nice people. Everybody really is passionate about combining theory and experiment. And so if you're interested in kind of doing theory in a way that interfaces very well with experimental neurobiology, I strongly encourage you to check out various labs we have at Genelia. And we also have a theory fellow program, which will give people the opportunity to kind of float between labs and develop your own research program within the Genelia environment. And then finally, I motivated this with zebrafish, but I only talked about one project. So I just want to mention that I'm lucky to have a large number of collaborators on different projects in zebrafish. I've just listed them here. And although they didn't necessarily contribute to this exact project, interactions with all these individuals have played a huge role in kind of shaping the types of problems I'm interested in terms of understanding kind of neural network mechanisms of behavior and activity throughout an entire functioning brain. And with that, I'll stop and take any questions.